Good afternoon, my dear friends. Today, I would like to present you the first lecture by normal physiology by a discipline called normal physiology under the topic Physiology of Excitable Tissues. First of all, let me introduce myself. My name is Madam Olga Moskovchuk. I am an associate professor of normal physiology department, and the subject you will study during this year over 3 modules till the middle of April called, as you understand, normal physiology. First of all, let me say that the subject matter of normal physiology is the foundation of the practice of medicine, and studying of its principles is essential for the practicing physicians. As an introduction into our subject, I want to show you the first slide, which will tell you about the very important things about physiology as a science. So, what this term physiology means? Physiology, translated from Greek as a physis, nature, and logos, word, study, research, science, is simply speaking the science about the nature of an organism. So physiology is as is if anatomy, while anatomy, human anatomy, is the study of the body's structures. The human physiology, normal physiology, is the study of how those structures work in a healthy organism. So physiology is scientific study of functions and mechanisms of their regulation in a living system. As a subdiscipline of biology, it focuses on how organ systems, individual organ cells, and biomolecules carry out their chemical and physical functions in a living system. One more time, I should repeat you that physiology is the scientific study of functions and mechanisms of their regulation in a living system. So those functions, as you see here, you may see the definition of the normal physiology, what this science studies, the science about the functions. Functions, what is function? Function is the specific activity of organ or system of organs. Each quantitative characteristic normally can fluctuate in some range, and this quantitative characteristic of each function called physiological parameters. So these functions, which are studied by physiology, can be evaluated by quantitative characteristics, which normally fluctuate in the normal range. Here you may see the definition of the norm. Norm is an average statistic value of the physiological parameter, quantitative fluctuation of which are able to keep the functional system of an organism at the level of optimal activity. And as you know, I hope that you know, that there are two basic principal mechanisms of all body functions regulation. There are nervous mechanism, which deals with conduction of nerve impulses through the nerve, and based on reflex principle, we will discuss this reflex principle in details during our practical classes. And second, humoral mechanism, humoral regulation, which depends on biological active substances carried by body fluids, mostly by blood. So physiology studies functions of a living organism, and mechanisms by which this function are allowed to adapt the human body to the changeable, to the variable conditions of the external environment, in order to keep the internal environment of an organism, internal environment, at stable level. That was just like introduction into the physiology as a science. Before starting a discussion of the physiology of excitable tissues, let's recall some basic things from the physiology of cell. And let's recall the structure of the cell and cell membrane. The cell is the structural and functional unit of the organism. The cell contains an outer membrane called plasma membrane or plasmalemma, a nucleus, the central structure of the cell, and a cytoplasm in which are cells organelles suspended in fluid and inclusion bodies containing secretion and storage substances allocated. The plasma membrane controls the rate and type of substances, ions and molecules passing into or out of the cell. Cell membranes are composed primarily of phospholipids and proteins. Let's first briefly discuss the phospholipid layer, lipid bilayer or biphospholipid layer. So phospholipids have a glycerol backbone, which is hydrophilic head. Look here, hydrophilic heads, polar and water-soluble hydrophilic heads, and two fatty acid tails, which are hydrophobic, non-polar and insoluble, and these tails face each other and form bilayer. That's why it is called biphospholipid layer. The lipid-soluble substances cross cell membrane because they can dissolve in the hydrophobic lipid bilayer. The water-soluble substances cannot dissolve in the lipid biphospholipid layer of the membrane, but may cross through water-filled channels or pores, or may be transported by special carriers. The second key element of structural organization of cell membrane are plasma proteins. There are basically two types of plasma proteins. The first type called integral proteins, which are embedded in the membrane, and peripheral proteins, which are located on either the intracellular and or extracellular side of the cell membrane. Functions of the cell membrane proteins are the transport of substances across the membrane. As you know, they form the channels for the ions pumps of the cell membrane. They also act as receptors. Peripheral proteins act as receptors, binding neurotransmitters and hormones, and by that changing the functional activity of the cell. And they also act as enzymes, catalyzing chemical reactions at the membrane surface. As you remember, there is also so-called glycocalyx layer, which is composed of glycolipids and glycoproteins. The functions of the cell membrane. The first function is that cell membrane organizes the structural support of the cell. Second function is transport of different molecules. Third function is receptive function. This structure of the cell membrane we have discussed earlier, as you remember from biology, called fluid mosaic structure of the cell membrane. So here you may see biphospholipid layer, heads and tails, yeah, proteins, integral proteins, surface proteins, and glycocalyx layer, which is made up of glycoproteins and glycolipids. As you remember from biology, the main physiological property of the cell membrane called semi-permeability, which means that it is selectively permeable for different substances. Now we are going to discuss the types of transport across the cell membrane, because this question is very important. It is a key question for understanding the functional activity of the cell membrane. The functional activity of each cell depends upon what substances can pass through it, in or out. Basically, there are two types of transport across the cell membrane, as you see here: passive transport and active transport. Let's first discuss passive transport, its main characteristics, and subtypes of passive transport. So, the passive transport, as you see here in this definition, does not require metabolic energy. It takes place down an electrical and concentration gradient, or so-called electrochemical gradient. Passive transport subdivided into few subtypes. The first, there are diffusion, osmosis, filtration. In some textbooks, you can find this last type, filtration, as a special type of transport across the cell membrane, which is not combined with the diffusion and osmosis. Yeah, but it doesn't matter. Yeah, you should understand this, that the main principles by which we combine them all together is that it does not require energy expenditure. Without energy expenditure, down electrochemical gradient. So, diffusion. Diffusion also subdivided into two types: simple diffusion, and facilitated diffusion. Simple diffusion. Small, uncharged, non-polar molecules can diffuse between the phospholipid molecules of the membrane, just along or down its concentration gradient. So such substances which can be transported by this type of diffusion, simple diffusion, are respiratory gases, carbon dioxide and oxygen, nitrogen, water, urea, ethanol, steroid hormones. Ions can move down both. Concentration and electrical gradient or electrochemical gradient. Because of their charge, ions cannot move between the phospholipid molecules. As you know, we have discussed a bit earlier that some membrane proteins, which are so-called integral proteins, can form in their structure water-filled channels or pores, which allow the passage of ions across the cell membrane, without energy expenditure. These ion channels are characterized by selective permeability, which depends on size of the channel, and the charges lining it. Size of the channel means the size of this filter of selectivity, and the charges which line these channels. Basically, there are three types of ion channels which are made up of the complex of plasma proteins found in the cell membrane. These types are voltage-gated channels. Here on the screen, you may see the structural organization of voltage-gated channels. Then there are also ligand-gated channels, and the last type, which is less numerous, called mechanically gated channels. So there are three types of channels, gated channels. Pay your attention. Gated. Gated means that they have special protein structures called gates. Gates. These gates will control the passage of substances, will permit or will limit the passage of substances through these channels. The voltage gated channel. So look, when movement of ions through the channels is altered by the membrane potential, it is called voltage gated channels. So it means that position of these gates, conformational changes which takes place, will occur following the changes of membrane potential differences. So on electrical, simply speaking, it depends upon the electrical state of the cell membrane. Other channels have receptors and are opened or closed by the binding of a hormone or neurotransmitter which alters the shape of the channel proteins. That's why these channels called ligand-gated channels. So an example of ligand-gated channels are cholinergic channels on postsynaptic membrane of chemical synapse of neuromuscular junction. Today you will see the structural organization of neuromuscular junction, and you will see these channels, ligand-gated channels on postsynaptic membrane and their role. Mechanically gated channels. So mechanically gated channels will respond on deformation of mechanical deformation of cell membrane. These type of channels are present in the cell membrane of Pacinian corpuscles. So let's go back. Yeah. So that was a short characteristics. One more thing, you should know that there is some type of channels which are so-called non-gated channels or leakage channels. These non-gated channels or leakage channels remain open permanently and also play their role in the stability of cell membrane potential, in maintenance of cell membrane potential. Let's go back. Here you will see that we have discussed only simple diffusion of non-polar substances, ions and molecules, and ions, charged ions, through the special ionic channels. Next type of diffusion called facilitated diffusion. Facilitated diffusion occurs down an electrochemical gradient like simple diffusion. It also does not require metabolic energy and therefore it is passive, but in compared to simple diffusion, it is more rapid than simple diffusion. So the rate of diffusion much higher than the rate of simple diffusion, and it is carrier mediated. So look, the solute to be transported binds to the carrier. There is special site for binding, which then the carrier changes its shape or conformational changes will occur, and by doing it moves the solute to the other side of the membrane. An example of carrier-mediated facilitated diffusion, we can give amino acid and glucose transport. Next type of passive transport called osmosis. As you know, I am sure that you know that osmosis is the flow of water across the semi-permeable membrane from a solution with low solute concentration to a solution with high solute concentration. The force that will limit extra passage of water through the semi-permeable membrane and which act against the flow of water called osmotic pressure. The presence of solute in the solution will produce this osmotic pressure. As you know, the solution with the high osmotic pressure called hypertonic, the solution with low osmotic pressure called hypotonic, and there is also isotonic solution, which have same effective osmotic pressure. The water will flow from the hypotonic to the hypertonic solution. And one more thing, that the part of osmotic pressure exerted by plasma proteins called colloid osmotic pressure or oncotic pressure. And these two parameters, let me say that osmotic pressure and oncotic pressure are rigid constants of homeostasis, and understanding of mechanisms of osmosis will help you to understand the processes of water exchange in an organism. And finally, the next type of passive transport, the last type of passive transport, called filtration. Filtration is the movement of water and solutes from an area of high hydrostatic pressure to the area of low hydrostatic pressure. The hydrostatic pressure developed by the weight of the fluid, and it is essential for movement of fluid along with dissolved substances from arterial end of capillaries into interstitium. And it also occurs in glomerulus of kidney. So one more time, let's repeat briefly. Passive transport, which does not require energy and takes place down electrical and concentration gradient, is subdivided into several subtypes. The first subtype, called diffusion, which also in turn subdivided into simple and facilitated, osmosis, and filtration. Active transport. The active transport generally is subdivided into two subtypes, primary and secondary active transport. How it differs from passive transport? It is clear that active transport, in compared to the passive transport, requires direct input of metabolic energy in the form of ATP, and in this case, the substances are transported against their electrochemical gradient. Generally, principally, there are two types of active transport, which are so-called, as I told already, primary active and secondary active, and there is also one special type, which also combined with these two types, because it also requires energy. It is called vesicular transport, in vesicles, transport in substances, in large molecules, antigens, for example, in vesicles. So, primary active transport. The primary active transport occurs against an electrochemical gradient. It requires direct input of metabolic energy in the form of ATP, and it is also carrier mediated. Examples of primary active transport are pumps, cell membrane pumps, sodium-potassium pump, here you may see that scheme, calcium pump, proton pump. So look, transport of sodium ions from intracellular to extracellular fluid and potassium from extracellular to intracellular fluid takes place against their concentration gradient in order to maintain low intracellular low intracellular sodium and high intracellular potassium concentration. The ratio is 3 sodium ions and 2 potassium ions. So each cycle will rotate 3 sodium ions against their concentration gradient out of the cell, because normally at rest concentration gradient of sodium ions directed here from here to there, from out to in, outside to inside, and 2 potassium ions from outside to inside against its concentration gradient. 3 sodium ions it binds here, 2 potassium ions it binds there on the outer surface, and energy requirements ATP hydrolyzed, and these ions will move in opposite directions simultaneously against their concentration gradient. By the way, this type of transport, according to another classification, also called counter antiport, so two substances simultaneously but in opposite directions. It is called counter antiport. Secondary active transport. So the secondary active transport is the transport of 2 or more solutes when they are coupled. One of the solute, usually sodium ion, is transported down electrochemical gradient and provides energy for the opposite transport of the other solute. Metabolic energy is not provided directly but indirectly from the sodium ions gradient, which is maintained across the cell membrane by sodium-potassium pump. As I told already, usually sodium is used, and that's why this type of transport called sodium-dependent transport. For example, glucose, sodium-dependent transport. First, sodium-potassium pump creates the high concentration gradient for sodium, and then sodium will provide this energy. The energy created for sodium will provide the energy of this gradient to transport glucose against its concentration gradient. And the last type of active transport called vesicular transport. So as you know, I am sure that you remember that vesicular transport subdivided into endo and exocytosis. Endocytosis is a transport of large molecules or particles in a membrane-bound vesicles through the membrane from the outside to inside or in opposite direction. And finally, we are going to begin our discussion about the physiological properties of excitable tissues. As you see here, nervous, muscular, and glandular tissues are said to be excitable tissues because they are capable, in response to stimuli, to rapid changes in membrane potential and to generation and action potential. All excitable tissues have a common and special physiological properties. The common or general physiological properties of excitable tissues are following. First of all, excitability, conductivity, refractivity, lability. These are common properties, general properties, and a few special properties, contractility and automaticity. What is excitability? What property called excitability? Excitability is ability to be excited. It is the ability of tissue to respond on stimulation by rapid changes of resting membrane potential. These rapid changes, reversible rapid changes of resting membrane potential, called action potential. The abbreviature AP means action potential. The abbreviature RMP called resting membrane potential. Remember this, please. RMP is resting membrane potential. AP is action potential. So, ability to respond on stimulation by rapid changes of RMP by generation of an AP called excitability. The process during which action potential is generated called excitation. Excitation. Next property, conductivity. These action potentials generated then are conducted along the nerve fibers, along the muscle fibers, from the nerve to the muscle cell, from the nerve to the nerve cell. So this ability to be conducted, to conduct impulses, called conductivity. Contractility is the ability of muscle tissue, exactly muscle tissue, not nervous and not glandular tissue, to change the length or tension in response on excitation. So excitation of the muscle cell finally results in contraction. Contraction means to change the length or to change the tension. Lability. Lability is the functional mobility of the tissue to produce impulses. So, simply speaking, it can be determined by the number of nerve impulses that are generated by the excitable tissue per unit of time. How many impulses are generated and then conducted. And based on this property, the nervous tissue and muscular tissue are characterized by much higher lability. Nervous tissue, the lability of nervous tissue ranges in between 1 to 3 to 4,000 hertz. It is lability, measure of lability, yeah, hertz, in hertz measured. The units are hertz. While muscular tissue lability much lower. It stays around several hundred hertz, so 2 to 300 hertz. One more common property of excitable tissues called refractivity. Here you may see refractivity. The refractivity, as you know, is when excitable tissue cell temporarily lose its ability to be excited. So during excitation, the cell temporarily loses its ability to be excited. It loses its ability to respond on stimulation by AP generation. And the last one, automaticity, is ability to self-generate an action potentials by the special cells, not all cells, as you know. There are special cells, high specialized cells in the cardiac muscle, the special subtypes of cardiomyocytes called pacemaker cells, heart conducting system cells, which have this ability to self-generate nerve impulses without stimulation. So we have discussed common and specific properties, the main characteristics of common and specific properties of the excitable tissue, and now we should discuss the what is resting membrane potential, the reasons and mechanisms of its maintenance. One more time, I should repeat you that RMP abbreviature means resting membrane potential. So resting membrane potential is the potential difference across the cell membrane. At resting state. At rest means not excited, not excited. Physiological rest means not excited. So, as you see, the value of resting membrane potential in the different types of excitable tissues ranges in between negative 30 to negative 90 millivolt. It is measured in millivolts, the units of measurement. In the nervous tissue, it stays in between 70 to 80 millivolts. In typical cardiomyocytes, around negative 90 millivolts. In atypical cardiomyocytes, much lesser, around negative 65 millivolts. In skeletal muscle cells, negative 90 millivolts. In smooth muscle cells, negative 30, negative 40 millivolts. In glandular cells, negative 20, negative 30 millivolts. What it originates from, yeah? So the resting membrane potential originates from concentration differences of sodium and potassium ions across the cell membrane, and which depends upon the permeability of the cell membrane to potassium and sodium ions. At rest, cell membrane of excitable tissues much more permeable to potassium ions, than to sodium ions. So potassium ions can diffuse out of the cell. When it diffuses out, it is called efflux or outflow of potassium occurs, making the inner surface of the cell membrane negative with respect to the outer surface of the cell membrane, which is a positive. But anyway, some sodium ions, small amount, but some sodium ions can also diffuse into the cell at rest along down its concentration gradient. So what mechanism will keep these potential differences, ionic asymmetry at constant level? So the concentration of potassium and sodium ions inside the cell are kept constant by the sodium-potassium pump or sodium-potassium ATPase, which actively, as we discussed already, actively transports potassium ions against its concentration gradient to potassium ions into the cell from here to there against concentration gradient, and sodium correspondingly sodium ions, 3 sodium ions out of the cell to balance exactly the diffusion of sodium into and potassium out of the cell. So this situation when the inner surface relative to the outer surface is negative charge, yeah, in compared to outer surface, it is negative charge, will produce potential differences across the cell membrane, which can be measured by the special microelectrode technique. One microelectrode inserted inside of the cell, another one placed into extracellular fluid outside of the cell, and at the very moment, the moment of pricking the cell membrane, the oscillograph will show us the value of resting membrane potential. In this case, as you may see, it is negative 70 millivolts. It means that the value of resting membrane potential 70 millivolts cell negative. So it is negative in compared to outside. Their action potentials. Now we should discuss the biopotentials. So the action potential is a rapid reversal of the resting membrane potential. Rapid reversal of the resting membrane potential called action potential, abbreviature AP. The action potential is the result of changes in the permeability of the membrane to potassium and sodium ions. As you remember, at rest, cell membrane was well permeable, perfectly permeable to potassium ions, which down their gradient move out of the cell because excess of potassium inside and excess of sodium ions outside at rest. That's why potassium moves out, sodium moves in, down their concentration gradient. Cell membrane almost, just a little permeable for sodium ions, just little sodium ions can diffuse in, yeah, but mostly cannot, because sodium ion voltage gated channels are closed, they are not activated, while it is well permeable to potassium ions, and potassium ions can easily diffuse out of the cell. This is situation at physiological rest. Cell permeable to potassium and almost not permeable to sodium ions. Now look, stimulation of the membrane causes opening of voltage-gated sodium ion channels, followed later by opening of potassium ion voltage-gated channels. As the membrane becomes less and less negative, more and more channels will open. Here you may see that value. Do you see this value? Negative 90 millivolts. This value is the value of resting membrane potential. The stimuli comes, the stimuli arrives. Depolarization begins. Depolarization begins because of opening of sodium ion voltage gated channels. Here you may see the first part of this first ascending curve, all this phase, all from here to there, called phase of depolarization, when cell membrane becomes less polarized than at rest. So it becomes less and less negative in compared to resting membrane potential value. Negative 90, negative 80, negative 70, 50, negative 50, and so on. Why it happens? Because positive charge sodium ions will influx of sodium ion channels through the voltage gated sodium ion channels happens, and cell membrane depolarization begins. If this level of depolarization reaches this point, the point called firing level or threshold potential or critical level of depolarization, there are different names, yeah, of this level: critical level of depolarization, firing level, or threshold potential. So if cell membrane depolarized and depolarization reaches this value, the fast sodium ion channels are activated. Those channels which I have shown you earlier, you remember, with two types of gates, activating and inactivating gates. Both activating and inactivating gates are open, and the sodium ions, rapid sodium ions influx occurs. The result of this is rapid depolarization phase. So the second part of this depolarization phase called rapid depolarization. Like it's like initial tilt threshold and then rapid depolarization. This point, so numeral 1 is depolarization phase. Then numeral 2, it is called overshoot. Overshoot is the portion of the action potential when the membrane potential is a positive, look here, positive 40 millivolts. Depolarization closes the inactivation gates of sodium ion channels and simultaneously to that opens voltage-gated potassium ion channels. Because of that, sodium ions influx through rapid sodium ion voltage-gated channels completely stopped, while potassium ion efflux through the voltage-gated potassium ion channels begins. The efflux of potassium ions will cause the phase which is called repolarization. Repolarization. So this numeral 3 will show us repolarization phase. The cell membrane will return to its initial electrical state. In some cases, there are two after potentials can be recorded. After depolarization from firing level to resting membrane potential. And after hyperpolarization from the resting membrane potential down, look here, negative 90 is resting membrane potential, it become even more polarized than it rests, negative 100, for example, millivolts after potential. But not always these after potentials are recorded. So one more time, the action potential is the rapid reversal of the resting membrane potential. The reason why it occurs is a stimulation. Now we will briefly discuss the characteristics of this stimulation. This stimuli should be threshold, minimal. Threshold means minimal but enough strong to depolarize cell membrane and to initiate an action potential. And also it should be adequate. The adequate stimuli for the nervous tissue, for the muscular tissue, is a nerve impulse. It is clear. The nerve impulse is threshold and adequate stimuli for excitable tissues. Properties of action potential are following. Action potential obey an all or none law. Action potential obey an all or none law. Action potentials are self-propagating, and during action potential, cell become refractory. So, as I told you, the first property is that it obeys an all or none law. Here you may see the definition of all or none law. Action potential arises in response to stimulation of threshold or supra-threshold intensity. Supra means higher than threshold. And its amplitude doesn't depend on trends of stimulation. So, simply speaking, if stimuli is threshold or supra-threshold, AP occurs. And there will not be more changes over amplitude of an action potential. But if trends over stimuli less than threshold called sub-threshold, AP doesn't occur. That is why it is called all AP occurs or none AP doesn't occur. But what occurs if the stimuli is less than threshold? The cell membrane will react on even sub-threshold stimuli. Now we'll see what type of biopotential will occur in that case. Let's go back here. So the graph of an action potential, phases of the of action potential. Here there are characteristics of action potential. It is characterized by lasting time, by duration. As you see in nervous tissue, it stays around 1 to 2 milliseconds. In myocardium, 300 milliseconds. And the amplitude. The amplitude is maximal deviation from the resting membrane potential. Look here. Look here, from negative 90 to positive 40 millivolts. So as you may see, the total amplitude of this deviation is 130 millivolts. Clear? 130 millivolts. On that slide, you may see the role of sodium and potassium voltage-gated channels in the origination of an action potential. Resting membrane potential and depolarization phase, and then repolarization phase, hyperpolarization, and again resting membrane potential. As you see, the depolarization occurs due to the activation, due to the opening of sodium ion channels, which are inactivated here at the peak of depolarization. These channels being inactivated because inactivating gates will close, but simultaneously to that, potassium ions voltage gated channels will be activated. And the potassium ions efflux from the cell will cause the phase called repolarization. Excessive efflux of potassium through the voltage gated and together with the leakage potassium ion channels can even cause hyperpolarization. Hyperpolarization means that cell membrane becomes more polarized than at rest. And one more time about characteristics of stimuli. The stimuli, according to the intensity, can be classified as threshold stimuli, which is minimal strength of stimulation that may initiate an action potential. And sub-threshold, the sub-threshold stimuli causes electrotonic potential or local response. Basically, I should say you that there are two types of biopotentials. The first type of biopotentials, as I told you, called an action potential, and I told you its characteristic that it obeys all or none law, that it is self-propagated, and during action potential, cell becomes refractory, while electrotonic potential or local response will differ from AP. Why? Because the local response to electrotonic potential, which is also rapid fluctuation of resting membrane potential. Look here, do you see this low amplitude deflection? Deflection. But it is not propagated. It appears some sodium ions influx occurs, but depolarization of cell membrane does not reach this value, and this potential will soon die out. So it like appears and disappears, and it does not spread along the membrane. So these are differences in between an action potential, spike potential, which spreads without changes in its amplitude, independently on distance traveling, and local potential, which is also very rapid fluctuation, but not propagated, but not propagated. Now we should discuss the changes over cell membrane excitability, of cell excitability, because of the changes of cell membrane permeability to sodium and potassium during different phases of action potential, which is followed by stimulation. So here you may see two graphs. This graph shows you the graph of action potential. That graph down there is the graph of refractory periods. Refractory periods. I remind you that refractivity is one of the common properties of the excitable tissues. So look, the first period of excitability, the first state of excitability, which is called normal excitability. Normal excitability coincides with the resting membrane potential. So when cell at rest, it is normally excitable. It means that subthreshold stimuli will initiate local potential, and threshold and suprathreshold stimuli will initiate an action potential. When depolarization begins during this phase of initial depolarization till threshold, the cell membrane excitability will increase. It increases, and this phase also sometimes called period of increased excitability, or exaltation phase, or supernormal excitability, because cell becomes more excitable than at rest. Then, as you know, the rapid depolarization phase, the fast opening and fast closure of sodium ion channels, and excitability of the cell decreases, do you see? And it takes place very fast, as fast rapid depolarization occurs. It drops till a period which is called absolute refractory period. As you see, this absolute refractory period coincides with the end of depolarization and first 2/3 of repolarization phase. Repolarization phase. So why this absolute refractory period occurs? Because of inactivation of sodium ion channels. Sodium ion channels are closed, inactivating gates will close, and sodium ions influx does not occurs more. If no sodium ion influx occurs, no stimuli, no strength of stimuli, no intensity of stimulation can excite the cell. Then, as you remember, simultaneously with the sodium voltage gated ion channels closer, potassium voltage gated ion channels open, and the result of this efflux of potassium ions, which will cause repolarization phase. During repolarization phase, excitability of the cell membrane gradually recovers. It recovers. And this period called relative refractory period. Relative refractory. It is not does not reaches its initial level of excitability, but it is not excitable already. So it recovers its initial state. What intensity of stimuli can cause AP generation? Super threshold. If super threshold stimuli applied, the action potential will occur. So this period coincides with 2/3 of repolarization phase. 2/3 of repolarization phase. And finally, as you may see, the excitability increases. It coincides to firing level. And here we see again exaltation or supernormal excitability from firing to resting membrane potential, from here to there. Second exaltation phase or supernormal excitability. Hyperpolarization will decrease ability to be excited, and this period called subnormal excitability. And then when resting membrane potential is recovered, it will be again normal excitability. So look, correspondingly to phases of action potential, how this ability to be excited will change. Look, first of all, resting membrane potential, normal excitability, initial depolarization, supernormal exaltation, or supernormal or increased excitability or exaltation phase, then absolute refractory period, closure of sodium ion channels, then relative refractory period, two-thirds of repolarization. Then again, supernormal excitability or exaltation phase 2, which coincides to this point from firing level to the resting membrane potential. Then hyperpolarization will give us subnormal excitability. And then finally, sodium-potassium pump will allow the cell to recover the initial ionic asymmetry. Ionic asymmetry, as I told you, is when potassium ions access inside and sodium ions access outside. And again, normal excitability. Now let's discuss some details from the physiology of nerve fibers. So the propagation of action potential in nerve fibers occurs by spread of local currents to adjacent areas of membrane, which are then depolarized to threshold and generate AP. So look how it takes place and what it depends on. It depends on the thickness of the nerve fibers, the diameter of nerve fibers. As you know, there are thick and thin nerve fibers. And it also depends upon myelination, the myelin sheath. So based on myelination and based on thickness, which will increase the conduction velocity. So look, the fiber size. First of all, fiber size, the diameter, thickness. Increasing diameter of a nerve fiber results in decreased internal resistance. Myelination. The myelin, as you know, acts as an insulator and increases conduction velocity. So, thick and myelinated fibers will conduct nerve impulses much faster than thin and unmyelinated fibers. And here in the table, you may see this dependence, this classification of nerve fibers into three types, A, B, and C types, called Erlanger-Gasser classification. And here you may see the functions of the nerve fibers, the diameter of the nerve fibers, thickness of the nerve fibers, and conduction velocity. And as you see, the fastest nerve fibers are A alpha somatic motor nerve fibers, which have the greatest diameter, which are thicker than the rest one, and the conduction velocity reaches 120 meters per second. In compared to C-type fibers, which are postganglionic, thin, unmyelinated, postganglionic fibers, which are thinner, and the conduction velocity correspondingly drops till 0.5 meters per second. So it depends upon the size of the fiber size, fiber thickness or diameter, greater diameter, faster. And myelination, if myelin sheath is present, faster than in unmyelinated fibers. Now look, the myelinated nerve fibers will show so-called saltatory conduction, while unmyelinated nerve fibers will exhibit so-called continuous propagation. These are just the names of types, mechanisms of nerve impulses propagation by myelinated and by unmyelinated fibers. Saltatory conduction in myelinated and continuous propagation in unmyelinated fibers. So here on that slide, you may see the general mechanism of nerve impulses propagation along the nerve fibers. So look, when action potential arrives, the sodium ions influx from extracellular to intracellular surface will occur. Yeah, so from extracellular fluid to exoplasma, exome. Inside there is exoplasma surrounded by exolemma. So sodium ions influx occurs. This sodium ions influx will bring more positive charged ions on the inner surface of the cell membrane, which, as you remember, at rest have a negative charge in compared to the outer surface. So here, as you see, the local currents occurs. So these sodium ions, which accumulated on the inner surface of the cell membrane, are redistributed along the cell membrane, bringing these local currents to the adjacent area of cell membrane, of exolemma. And now this area of cell membrane becomes excited. It becomes depolarized due to the local currents flow. Then depolarization reaches threshold firing level. Fast influx of sodium will occur and AP propagated from one area to another. And by that way, the local currents will flow along the nerve fiber membrane, bringing its positive charge, depolarizing, and initiation action potential generation. So this mechanism generally called the local currents flow, the mechanism of local currents flow. Pay your attention here that the area of part of the nerve fiber which was excited earlier now become repolarized. This red color is the area which is repolarized, and you may see that potassium ions efflux occurs. Potassium ions efflux causes repolarization. So the nerve impulses can be propagated along the nerve fibers only in one direction, normally in a healthy organism, only in one direction. Do you see? Because here, this area now in refractory period. It does not respond on stimuli. These local currents cannot excite this area because it is in relative, absolute, and then relative refractory period. Look here that scheme. This is a scheme which shows the mechanism of nerve impulse is propagation along unmyelinated fibers, which is called continuous propagation. The area which is excited, which was excited, the sodium ions in flux, the local currents will flow from excited to unexcited to the neighboring, to the adjacent area of the membrane, depolarizes this adjacent area of the membrane, yeah? Fast influx of sodium ions occurs and AP generated. And by that way, area by area continuously, slowly depolarized. And by that way, the nerve impulses will spread along the exolemma. Here you may see principally the same mechanism, which is called saltatory conduction, and show us the mechanism of nerve impulses propagation along myelinated fibers. As you know, in myelinated fibers, there are some gaps of myelin sheath, which are so-called Ranvier nodes. And here in Ranvier nodes, there is a lot of sodium and potassium ion voltage gated channels. So the density of these channels is very, very high. While here, there is no absolutely sodium and potassium ion channels, and because myelin sheath act as insulator, the local currents cannot flow here. So principal mechanism the same. The action potential arrives, depolarization of this Ranvier node occurs, local current will flow from one node to the neighboring, to the adjacent node, and now adjacent inactive node, which is at resting membrane potential, become also active because of accumulation of this potassium sodium ion channels, sodium ions in flux through the fast sodium ion voltage gated channels, and action potential occurs. And by that way, the action potentials will like jump from one node to another, from one to another. That's why this way of nerve impulses conduction along myelinated fibers is much faster than in unmyelinated fibers. The extracellularly recorded electrical activity in such a nerves reflect the total effect of all the individual action potentials in different axons and is called a compound action potential. When a nerve, like for example sciatic nerve, which consists of many bundles, many nerve fibers, which such a nerve is stimulated electrically, it can be shown that the amplitude of the compound action potential increases as the stimulus strength is increased. This observation simply reflects the presence of axons with different thresholds within the nerve. As the stimulus is increased more and more, axons reach threshold so that a greater proportion of fibers fire and action potential no further change. This type of potential are called compound action potential, the compound action potential. Because now we are discussing on the models. These models will show us one nerve fiber. But in an organism, yeah, the nerve trunks contain many nerve fibers. That is why you should understand what is compound action potential. Compound action potential. This compound action potential will reflect the total effect of all individual action potentials in different axons from which this nerve trunk consists of. The laws of conduction in the nerve fibers are following the law of physiological and anatomical continuity of a nerve. So the nerve should be intact physiologically and anatomically. The law of isolated conduction, I told it right now, yeah, the isolated conduction, the nerve impulses will spread by each fiber separately. The law of two-ways conduction, the law of conduction without decrement, and the law of relative infatigability. Synapses. The synapse is the site of transmission of electric nerve impulses between two nerve cells or between nerve cell and muscular cell or between nerve cell and glandular cell. The structure of synapse, chemical synapse, first of all, yeah, the synapses are classified into chemical and electrical synapses. The majority of synapses in CNS are chemical synapses. So here on that scheme, you may see the structure of the chemical synapse. Why chemical? Because the signal transmitted is a chemical neurotransmitter. There are also electrical synapses mostly in the central nervous system. The synapse, according to the effects, can be classified into excitatory and inhibitory. If it causes excitation of postsynaptic membrane, excitatory, and correspondingly inhibitory if postsynaptic membrane is inhibited. According location, peripheral and central. And the chemical synapse consists of presynaptic membrane, postsynaptic membrane, and in between them synaptic cleft. Presynaptic membrane is axon terminal, yeah, you see here, axon terminal with a calcium ion voltage gated channels. The postsynaptic membrane contains ligand-gated channels. So look, how this synaptic transmission, the transmission of nerve impulses through the chemical synapse happens. Step number 1. The action potential arrives at a nerve terminal over axon. This action potential will depolarize the presynaptic membrane. Depolarization causes opening of voltage-gated calcium ion channels, and result of this, calcium ions influx will occur into presynaptic membrane. The calcium ions influx in turn causes release of neurotransmitter from the vesicles into synaptic cleft by exocytosis. So these are synaptic vesicles. These vesicles contain the molecules of neurotransmitter. It fuses and releases by exocytosis neurotransmitter into synaptic cleft. The neurotransmitter diffuses across to the postsynaptic membrane and binds with the postsynaptic receptors. And now the result of this will depends upon what electrical state will occur on postsynaptic membrane. The excitatory neurotransmitter depolarizes the postsynaptic membrane, bringing it closer to threshold and closer to firing and action potential, to the firing level, to critical level of depolarization. The resulting action, the resulting biopotential, which will occur on postsynaptic membrane, called EPSP, excitatory postsynaptic potential. The excitatory postsynaptic potential is not an action potential. It is local electrotonic potential. But it will bring the cell membrane electrical state closer to threshold and closer to firing and AP. Another variant, when inhibitory neurotransmitter are released and hyperpolarizes the postsynaptic membrane, moving it away from threshold and further from firing and AP. And as a result, the inhibitory postsynaptic potential occurs. The abbreviation is IPSP. Here, look, you may see the two graphs. EPSP, do you see these local potentials, electrotonic potentials? This is threshold of firing level. So each EPSP, they move electrical state of cell membrane closer to the firing level, to the threshold. And as a result, AP occurs. While IPSP, caused by hyperpolarization, will move the electrical state of the membrane far from the resting membrane potential. Hyperpolarization of the cell membrane. What it depends on? It depends upon ions. You know it very well, yeah? So if sodium ions influx of sodium ions will cause EPSP. Efflux of efflux of potassium ions, efflux of potassium or influx of chlorine ions will cause hyperpolarization of the cell membrane. So what it depends on? It depends on what channels, ligand-gated channels on postsynaptic membrane are activated, following binding the receptors with the neurotransmitter released by presynaptic membrane. So what will cause AP generation if both are local potentials? Summation. So what happens? Summation over EPSP, summation of these local potentials. Each local potential make it more and more and more depolarized. So spatial summation, if many knobs simultaneously are excited, simultaneously, and depolarization reaches this firing level, or temporal summation, when this excitatory postsynaptic potential will occur repeatedly, one after another, repeatedly. This also will cause summation of EPSPs. The cell membrane depolarization reaches threshold, reaches firing level, and AP will occur. So that is the mechanism of synaptic transmission. And now a few words about physiology of muscles. As you know, there are skeletal muscles, which are so-called striated muscles, smooth muscles, which are so-called non-striated muscles, and cardiac muscles, special type of muscles, supply our human body. Each muscle fiber is a multinucleated single unit. Yeah, do you see? This is muscle fiber. Multinucleated single unit, single cell. It contains bundles of myofibrils. Do you see? These are, this is muscle fiber, and this is myofibril. Bundles of myofibrils surrounded by sarcoplasmic reticulum. Each myofibril contains interdigitating or overlapping, look here, interdigitating or overlapping, do you see these areas? Thick and thin filaments or myofilaments that are arranged longitudinally in the sarcomere, A sarcomere runs from Z line or Z membrane to Z membrane. Here you may see a zone of overlapping. In the center of sarcomere there are thick myosin filaments, and here there are thin actin filaments. Uh, Thick filaments contain myosin. Each myosin molecule has two heads attached to a single tail. The myosin heads bind ATP and actin and are involved in cross-bridge formation. Thick filaments are present in the A-band, you see here, A-band, in the center of the sarcomere. Thin filaments. Thin filaments contain actin, tropomyosin, and troponin. Troponin is the regulatory protein that permits cross-bridge formation when it binds calcium. Thin filaments are present in the I-band and interdigitate with the thick filaments in a portion of the A-band. T-tubules, you see here, yeah, transverse tubules, T-tubules. T-tubules are an extensive tubular network open to the extracellular space, which carry the depolarization to the cell interior and are located at the junctions of A-band and I-bands. Sarcoplasmic reticulum. The sarcoplasmic reticulum is the internal tubular structure that is the site of calcium ion storage and release for excitation, contraction, coupling. It has terminal cisterns that make contact with the T-tubules. Their membrane contains calcium ATPase pumps, which transport calcium for intracellular fluid into the sarcoplasmic interior. Steps over excitation, contraction, coupling. Excitation, contraction, coupling will show us how excitation of muscle fiber initiates its contraction. So look, by the way, the nerve impulse, you should know it, the synapse in between axon terminal of motor neuron and muscle fiber, the membrane of muscle fiber, sarcolemma, called neuromuscular junction. Neuromuscular junction. So the steps of excitation and contraction, excitation, contraction, coupling in skeletal muscle. Action potentials in the muscle cell membrane initiate depolarization of the T-tubules. The depolarization of the T-tubules opens calcium ion channels in the sarcoplasmic reticulum, causing release of calcium from sarcoplasmic reticulum into the intracellular fluid. Here you may see the cisterns of sarcoplasmic reticulum, yeah, and here you may see the transverse tubules. Intracellular calcium increases, you see, yeah, this like pepper granules, yeah, it is intracellular calcium ions. Calcium ions binds to troponin C, it binds to troponin C on the thin filaments, causing, here you may see, here you may see it, causing a conformational change in troponin. Because of that, tropomyosin is moved out of the way so that the cross-bridge cycle can begin. Actin and myosin bind, the head pivot, the thin and thick filaments slide over each other, and ATP is hydrolyzed. The muscle can shorten up to 60% of its resting length. Subsequently, the cross bridges break and a new molecule of ATP binds to the myosin head so that a new cycle can begin. And the final stage is relaxation of the muscle that occurs when calcium ions uptake into the sarcoplasmic reticulum will occur, and the concentration of calcium in intracellular concentration of calcium decreases. So these are steps of excitation, contraction, coupling, which will show how excitation will initiate contraction of the muscle. The trigger of the muscular skeletal muscle contraction is calcium ions, which are released from the lateral cistern of sarcoplasmic reticulum and which binds to troponin C and expose active sites on actin molecules and will allow the formation of cross bridges. Types of muscular contraction is subdivided into single twitch or single contraction and tetanic contraction. And there is also one more classification: isotonic, isometric, and auxotonic contraction. Look, an individual stimulus causes single contraction or single twitch. There are first period of single twitch called latent period, the period in between stimulation and beginning of contraction, which lasts 0.01 second. Then next phase, this ascending curve will show us contraction phase, 0.05 seconds, and which is followed by a relaxation phase, 0.05 seconds. Skeletal muscles receive a number of nerve impulses, not single signals from the central nervous system. And result is the intense and continuous contraction of the muscle, which is called tetanic contraction. Look here, or tetanus. The tetanus or tetanic contraction is the result of summation of single twitches. Dependently on the phase when the new impulse appears, the tetanic contraction can be classified as complete tetanic contraction, complete, if the next stimuli, the new stimuli, arrives during contraction phase, or incomplete tetanic contraction if the new stimuli arrives during relaxation phase. So thank you for your attention. I hope this material was useful, but the rest details, we did not discuss many details, and we can detail them all together during our practical classes. Thank you for attention. Goodbye.